All the events are, all the sessions are very good.
Thank you, sir. Thank you for complimenting, and thank you for accepting the invite too, sir.
Pleasure, ma'am.
Within a minute or two, we can start our program, sir.
Yes,
If
ma'am.
still
Yes.
participants are joining, we can give 2 or 3 more minutes time to join.
No problem.
Then we can start, sir.
So, how many participants are there daily? For all the
Sir, daily on the chat we can see 80 participants, sir, but when we take the attendance reports, we are getting almost 120 to 130 participants. Like there will be a few participants who are in and out, so all that tally goes, and finally we are listing.
who are the participants? Means like they are faculty or they are PG students?
No PG students. See, this short-term training program is exclusive for the faculty members. So no students, and the other rather like UG or PG is not at the time. So only the faculty members, and especially we have mostly the 40% of professors are there, and the rest 30% are with associate professors. So mostly a lot of doctorates are registered.
Okay, okay.
So we kindly mute the participants. Please check the mute list. Yeah, how about the things there in sir? Babatla, everything fine?
Everything is fine, ma'am.
Okay.
So you're from pharmacology background or from other?
No, I'm from pharmaceutics background, sir.
Pharmaceutics, okay.
Yes, sir. Yes, sir. I'm from pharmaceutics. Sir, with your permission, can we start, sir? It's almost here with a number of 45 participants in the line, and probably they may join within 5 or 10 minutes.
Yes,
so
ma'am, I'm ready.
We can start. Yes, sir. So, very warm good morning to all the respected faculty members. And today we are into the day 3 of this AICTE program organized by Sun Hills College of Pharmacy and sponsored by AICTE. And we are into this phase 3 of this STTP on preclinical and clinical aspects in molecular neuroscience. And I hope you all had a wonderful days of day 1 and day 2. And we are believing that the objectives of this STTP is being partially fulfilled, and we trust that it's been completely a better one by the end of a day 6. So we are into this day 3, and today we have our expert speaker, Dr. M. Satish Kumar sir, and sir is an IQAC coordinator and HOD of Department of Pharmacology, Bapatla College of Pharmacy, Bapatla. And today, sir is going to deliver a talk on behavioral neuroscience research and the trends in Andhra Pradesh. Sir, it's an immense pleasure to have you on this platform, and also I thank you for accepting the invite. And to do the honorary introduction, I request my dear colleague, Mrs. Shivagami Madam, to just introduce the achievements of Dr. Satish Kumar sir to respected faculty members. Madam, it's over to you, and kindly do the meet. Thank you.
Yes, madam. Yes. Good morning, one and all. We welcome you all for today's session. I'm Mrs. B. Shivagami, working as Associate Professor in the Department of Pharmaceutical Analysis. Today, I'm very proud to introduce today's speaker, Dr. Satish Kumar. Dr. Satish Kumar is IQAC Coordinator and Head of the Department of Pharmacology at Bapatla College of Pharmacy, Bapatla, Andhra Pradesh. Dr. Satish Kumar received his PhD in Pharmacology at Acharya Nagarjuna University, Andhra Pradesh. Sir completed his M.Pharm in Pharmacology and B.Pharm from Tamil Nadu Dr. MGR Medical University, Chennai. Before joining the faculty at Bapatla College of Pharmacy, Sir was a program coordinator for pharmacy and health program at Texila American University. Sir was an accreditation panel expert for Higher Education Authority, Ministry of Higher Education, Republic of Zambia. Sir has received in university bodies and committees such as Governing Council, Board of Studies, Academic Program, Accreditation, Research Ethics and Quality Assurance. Sir is a recipient of 3 national awards for his research in malnutrition and industrial effluent management. His research interests lie in the areas of preclinical neuropharmacology and pharmacoepidemiological studies. Sir has authored quality research papers and book chapters in the area of preclinical and clinical pharmacology. Sir has so far received 4 research seminar grants from UGC, AICTE and ICMR. Sir has experience and expertise in accreditation and academic audit related activities of higher education institutions. Thank you. I would like to hand over the session to Dr. Satish Kumar, sir. Sir, please take over the session, sir. Thank you.
Thank you very much, ma'am.
Thank you, sir.
Thank you, Shivagami Madam, and welcome to you, sir. We just continue with the session.
Yes, ma'am. So I hope you are able to see the screen. And my voice is audible. Yeah.
Yes, sir. Yes, sir. Perfect. We can continue.
So I would like to thank Seven Hills College of Pharmacy for giving me the opportunity to speak on this particular AICTE sponsored short-term training program on molecular neuroscience. And I have watched all the 3 sessions on the first day of this 3rd phase. It was very interesting and it was very useful. I attended 1 session of yesterday, and I have chosen a particular topic that is on behavioral neuroscience research. What is the current trend in Andhra Pradesh? What sort of research is going on? Who are the researchers who are involved in this behavioral neuroscience research? And that too specifically on preclinical side. So though I have given the title as behavioral neuroscience research, the contents will be all related to preclinical side, not to the clinical side. So as you can see in this slide, I'm the IQAC coordinator as well as I am the HOD of Department of Pharmacology at Bapatla College of Pharmacy. And Okay, so let me straight away go into this presentation. So if you ask me whether I'm an expert in this particular topic on molecular neuroscience or its related areas, my answer is no. I'm not an expert. I'm still a beginner and I'm working to improve my knowledge or skills in this particular area. So what is that which is very much related to this particular topic? What skills I have or what is that I have done so far in this particular area? In my master's, I had an opportunity to do my master's project work in the Torrent Research Center, Gujarat, and I was a trainee there. During that time, they were developing animal models for subarachnoid hemorrhage. So most of the faculty members who are attending the sessions who are interested in this particular area, you might be knowing in stroke, the most common type is cerebral ischemia. And most of the preclinical research will happen towards that particular side only because 80% of the stroke cases is related to cerebral ischemia. Whereas there is another 20% in stroke, cerebral stroke. That area is called as hemorrhagic stroke. So the rare opportunity what I got during my master's time is that I was involved in the development of animal models for subarachnoid hemorrhage. Subarachnoid hemorrhage, which is usually around 5 to 7% of the total cerebral stroke cases reported. And the name of the model is itself is related to that. Most of us will be knowing about the middle cerebral artery occlusion. That is for cerebral ischemia. And I had an opportunity to work with middle cerebral artery perforation. Perforation where we can cause bleeding into the brain tissue and then we can create cerebral hemorrhage or subarachnoid hemorrhage. So that was one aspect which I am connected to this particular topic. So I have a very hands-on experience on developing cerebral hemorrhage or subarachnoid hemorrhage animal models, maybe around 16 months. And our job was also related to that. And after that, when I came here to Andhra Pradesh to do my PhD work, so I wanted to increase my skills in this particular line only. So in my master's also, I took stroke only as my major area of research. And even in that one also, instead of concentrating on cerebral ischemia models, I did my PhD work on subarachnoid hemorrhage model only. So that is the another important phase where I work more in deep in this particular area. So that is my second connection with this particular topic. So for more than 6 to 7 years after my PG, again I worked on hemorrhagic stroke. And after that, after this PhD work, we established this behavioral research lab, especially related to Alzheimer's, and specifically concentrating on the instruments which are related to the in vivo studies for learning and memory or nootropic activity. So we had some around 7 to 8 equipment or instruments which are related to learning and memory process, different types of mazes. And uh, in 2 institutions where I have worked and presently where I am working, in both these institutions, I had that interest of developing a basic behavioral research lab with emphasize on screening of drugs for learning and memory activity. And the area which I'm consistently trying is to get a smart video tracking system instead of going for a semi-automatic one or the manual mode. We wanted to make it as a complete one by having a smart video tracking system. But personally, I have a target that I need to get that instrument only through grant, and I don't want to make my management to spend for purchasing that equipment, though it is costing around 3 to 4 lakhs approximately if it is an Indian model, and if it is a standard one from a international manufacturer, it could be a few more lakhs more. But my target is whether it is an Indian video tracking system for mazes or whether it is an international brand. I wanted to get that particular video tracking system only through the help of funded projects. So for that, I'm waiting and I'm consistently writing grants related to that particular area. So this is the brief introduction of how I am associated with this particular topic. And again, I'm telling you, molecular neuroscience area is a very bigger one, and it's an ocean, and only I am very much related to Alzheimer's, and next to with stroke, and mostly with users of mazes, how to screen the nootropic effect of drug substances. That is my area of research. Okay, so this picture, what you are seeing, is something to say that every one of us is just a beginner. No one can say that I'm an expert or I'm excellent in this particular area, and that's the reason why I hear that right from the senior faculty to the youngest faculty, whoever is related to this particular neuroscience, whether they are from pharmaceutical analysis or whether they are from Sutix or from Pharmacology, whatever department, whoever is interested in these areas, molecular neuroscience, that's why you are attending, and that is the reason why I am also listening to these sort of presentations. Okay, so whatever I'm presenting here, it's all not the expert views. It's just my personal views, and whatever knowledge I have gained, I'm just sharing with you people. Okay, so allow yourself to be a beginner, and no one starts off being excellent. That's the theme. And what is that I want to talk about? I have 4 things to discuss with to all the participants. The first one is that what are the things we need to learn from the experts? This is a general idea, and I want to give 3 case studies. And these 3 case studies I actually took from the first 3 sessions of this program. So I listened completely to all the 3 sessions of day 1 of this phase 3 of this AICTE sponsored STTP program. And from that, I developed 3 case studies, which I want to share, or what I have understood about their presentations. And the third important aspect is something which is relevant to my topic. What is the actual trend in this molecular neuroscience, especially in preclinical research in Andhra Pradesh? And along with this particular theme, I also want to highlight how can we find an article, or how can we find a potential collaborator for our research which is related to molecular neuroscience. So that will be one of the core aspects of my today's presentation. And of course, because we are speaking all these things related to research, I just wanted to give a simple checklist taken from an article, and I have modified that checklist of the research proposal so that whoever wants to do any sort of preclinical research related to molecular neuroscience or neuroscience in general, they can use this checklist. They can get themselves prepared for writing a research proposal, not only for any funding agencies, even for their PhD purpose as well. So these are the 4 aspects which I will be discussing it today, and I'll go to the next one. Now, this is something which I want to share. This is from my understanding. This is my personal view. And we all are beginners. And I told you, we are not experts in this particular field. And therefore, as a beginner, we have to always listen to the experts who have who have already proved themselves in this particular field. So with that point of view, I have been working for the past 13, 14 years, and even on the day one, when I listened to all the three sessions, I was just looking curiously to those experts, what point they are sharing, what is that new thing which I am trying to get out of their presentation. So actually, I took the lead from their three presentations. So through this particular slide, I want to introduce to the participants what is that we need to know from the experts. So I'll start from this one in this cycle. So let us start with the first one. Let us identify something. What is that we need to identify? First of all, we have to identify who are the experts related to my research area. So I need to identify who are those experts, how to find them. So some 2, 3 years back, we used to attend seminars. We used to go for some national or international level conferences. We used to attend workshops, something like that. So we go there, and then whenever we participate there as a delegate, we will always like to see who are the resource persons, where are they from, which type of organizations they are representing, whether they are representing the government research laboratories, whether they are representing the private companies, or whether they are representing the universities, whether they are representing some of the leading private pharmacy colleges or institutions. That's what we always look. And after seeing their names and their associated organizations, we always have that curiosity to know what sort of research they are doing and what is the significance of their institutions. So this is how we actually identified the resource persons or the experts in our area. And because of COVID, suddenly there is a huge opportunity for all of us. And from last March 2020 to this June end, we had enough number of lectures from the experts, and we had an opportunity to listen or see them through our mobiles. And of course, we can say that during this 2021, we have almost become tired of listening to so many lectures. So that many number of lectures are happening, and now we are able to choose which one we need to attend. Like that many opportunities are there, but to be remembered, we cannot neglect anyone's contribution. So as much as possible, we have to find some time and then to listen to at least to those lectures, or we need to attend those sort of trainings, online trainings, which are relevant to at least to our area of research. So first of all, we need to identify our research area. Then we need to identify who are the experts in that particular field. And we have to identify where all these experts coming from, whether they are representing government, whether they are representing the companies where we are trying to find jobs, whether they are from academic institutions or whether they are independent researchers. So we have to look out for all these people. So then after identifying the area of research and the institutions who are doing that sort of research, then what we need to do? The next thing I will recommend is that we have to start listening to them as much as possible. Then we have to follow them. We have to follow them. Follow them in sense, because of COVID and because of other limitations. Following means not directly following. So we have many ways to follow them. And one of the best tools to follow is to effectively make use of the social networking sites, especially Facebook and LinkedIn. These sort of social networking sites, we can effectively make use of it, and then we can either get to know about them, get the updates, what they have been doing or what they have been publishing, and also follow them continuously. Now and then, whenever they do some achievements, whenever they impress us by something, then we always just keep a message that, okay, sir, good work. You are inspiration for us. Like that simple message. We can always put to them, not just wishing them on birthday, not like that. At least professionally, we can follow them. And once we start listening and then following them, then if we get an opportunity, then try to meet them. So again, the opportunity now is to either to reach out them by using WhatsApp, by uh, talking with them through the other social media platforms. We can always do, and then we can uh, follow that. And after meeting them through direct or indirect mode, that is online or offline mode, then always ask them about what is special or what needs to be done in that particular area, and whatever doubts you have on that particular area, or what sort of an expert advice you want to get from them. So ask everything, right from basic to the most advanced questions. Whatever you have, you ask everything to them, and learn from them. So by whatever means they are giving their research outputs, try to follow each one of their research outputs. See what is their patents, see what is their research publication, see what is their research presentations. So like that, there are different means of research outputs, book chapters, books written by them. Okay, so try to follow all their research outputs and try to take the inputs from them. Of course, when I say this COVID has given more opportunity for us to follow them online, but we cannot neglect the direct hands-on person-to-person interaction which we have been doing 2 years back. So it's like two equal opportunities. Whenever you get an opportunity to meet them, ask them, or learn from them directly, when you are able to learn by hands-on, discuss certain things person to person, of course that will be the most perfect opportunity to learn. But because of any reasons, when we don't have that opportunity, and because of some limitations, we are only able to meet online, still make use of those platforms very effectively, and then try to get their expertise. So meet, ask, and then learn from them. This is the next strategy. And now, once you meet them, ask them, learn from them, and once you show your commitment to them, once you showcase your skills to them, so somehow you try to impress, and try to make sure that you do some sort of collaborative research work with those sort of experts. When I say experts, of course, without any levels or priority, it takes government research institutions, it takes the leading private universities, and then private colleges, accredited institutions. And it's not just related to pharmacy alone. We need to understand, whenever we have a research interest, or whenever we are towards an area of research, it's not just only the pharmacy related institutions are doing this. We can always collaborate with those people who are from other fields also. For example, you can relate with arts and science people, you can relate with engineering people, we can relate with agricultural people, we can relate with medical people. And since it is preclinical research, we can always collaborate with veterinary people. Like this, many fields of researchers are there. So we can collaborate with everyone and not limit ourselves only with the pharmacy researchers. So this is one particular thing which I want to share here. So we need to collaborate with these sort of experts, and after collaborating, not just doing some research, we need to showcase our research output. We have to bring out something. We have to bring some research output. It can be anyone. It can be a publication. it can be a presentation, it can be a patent, it can be a product, it can be an exhibit, it can be a book chapter, it can be any form. It can be a video tool for providing an awareness for the other fellow researchers. So try to bring out your research output in any form, in whatever way you are very much comfortable. Only when you showcase your research output to the outside world, then you also get the opportunity to work with many more experts. You can always continuously improve your skills. Otherwise, you will get satisfied with whatever you learned or whatever you have, and then you will keep quiet. It's not that way. We have to always improve in each and every step. Okay, so collaborate, and after collaborate, you try to showcase your research work in the form of publications, presentations, or patents. And the last one, you identified, you followed, you learned, you collaborated, and you showcased yourself. Then is that sufficient? No. What is that we need to do? Irrespective of your experience, whether you have learned a simple thing or whether you have learned the most advanced thing, whatever may be, you try to always share it with other people. So it's not that you have to wait until you become an expert and then to go and share something with others. It's not that. Even you have learned a very small thing, a very basic thing, but if you know that this basic can be helpful to someone, don't think that, don't hesitate or don't feel shy that I'm not an expert, so why should I share this? Will they consider my advice or will they consider my information? Don't take it in that way. Whether they take it or not, if you have an opportunity, go and do it. So the last important aspect is try to motivate people. Try to motivate people. Try to teach someone. Try to train someone. Try to provide some hands-on experience to someone. And if you are not able to motivate, if you are not able to teach or train something, at least support them morally, okay? So I'm there, I'm interested, at least saying that I'm listening to you. When you say that I'm listening to someone, they'll feel happy, they will try to express themselves. So motivate, teach, train, and try to support in whatever way possible, especially the students and the young researchers in that particular field. So when you do that, automatically you also learn from them and you share your experience with them. So this cycle is not something which starts with identifying area of research or identifying the experts in that field. It doesn't start there and then end with motivating, teaching, training, or supporting the rest of the people. It doesn't stop there. It's a cycle. This cycle has to run continuously until we breathe the last breath in our life. This cycle has to go on. If it goes on, at some point of time, you will feel happy that I have done something, I have achieved something. And like that, at least we will get self-satisfaction at the end. Okay, so this is a custom-made diagram from my experience or the information which I want to personally share with my fellow participants. Okay, now, as I was telling, my second agenda or my second concept is that I have listened to all the three presentations of the first day of this STTP phase 3, and I told you that I want to make some case study from their three presentations. Of course, I have not taken their permission. I am not supposed to use their data without their support, but at least I consider that because it is for educational purpose and it's of course for motivating the other people. So I want to highlight some of the important features about those researchers who spoken on the first day of this STTP. So my first study will be with the help of Dr. M. Ramanathan sir, who was the resource person on the first day. And he's the principal of PSG College of Pharmacy, Coimbatore. And I want to highlight some points here. Okay, so I know him, though he doesn't know me, but I know him, I follow him. And when I looked into this PSG College of Pharmacy website, then I found that, okay, so especially sir, who is also the HOD of that department. The Department of Pharmacology in PSG College of Pharmacy is focusing on neurosciences and neurodegenerative disorders, and they are giving also equal importance to cancer research. So which you can always find from their website. And I have seen that they have got many funded projects for their institutions altogether. But Ramanathan sir alone, means as a investigator or in the capacity of a co-investigator, he himself is involved in different capacities for around projects worth more than 4.25 crores. So it may not be accurate. I have taken some figures from their website. So it has to be more than this, maybe not the updated one. It has to be more than 425 crores worth of funded project Sir has done as an investigator, as a co-investigator related to neurosciences, neurodegenerative disorders, and cancer. And this institution also has some of the quality research instruments. And one of the most impressive one for me is the stereotactic device. So the other name is stereotaxic. Some companies call it as tactic and some call that as stereotaxic. So what is the use of this particular one? It's mainly used for the stroke research, especially when you want to damage a particular part of the brain tissue. So you can directly drill through the skull of the rat or the rodents, and then you can inject the rodent's blood, its own blood, and then you can cause some sort of a hemorrhage, or you can cause some sort of an ischemia or vasospasm in the brain tissue of the animals. So a very branded a stereotactic device from RV or WD Life Sciences. And there are many other instrumentations, ELISA, reader, PCR, and many other for histopathological studies and other things. So this is one thing which impressed me. And I, of course, it's not for this presentation. I already know about the facilities available in this institution because one of my good friend was working earlier in this particular college, and it's one of the premier institutions there. And one thing which I wanted to highlight from Sir's presentation is most of us who are involved in preclinical research, we would like to always concentrate on in vivo models, and very less number of people, they get involved with in vitro models, in vitro preclinical models. So Sir was expressing that they have the facility for performing both in vitro models as well as in vivo models, and he was highlighting this particular point there. It is OGD/R, which is oxygen glucose deprivation with reperfusion model. And in vitro technique, of course, we need some sophisticated equipment for that. And he was also explaining some of the in vivo models with an abbreviation of BCCAO. And also some researchers refer it
காத்தே
as.
வரலப்பா உள்ள உட்காரவே முடியல கரண்ட் பவர் கட் இன்னைக்கு ஃபுல் பவர் கட்டப்பா தெரியல
I think someone.
Sorry for that, sir. Some participant has turned on the mic. That's it.
Okay, no problem. Okay. So he was expressing about in vivo models, and he was also speaking about cerebral ischemia. And from their publications from their website, we also know that in cerebral ischemia, they also do middle cerebral artery occlusion models as well. So one important point I learned, or which I want to emphasize to my fellow participants who are listening to me, is that try to identify the opportunities for in vitro, standard in vitro techniques for screening of drug substances. In this case, maybe OGD with reperfusion. So this one particular point I wanted to highlight. When you want to go for a quality publications, when you do with these sort of molecular level studies, or when you do with these sort of in vitro based studies, there is a very less competition, and there is a very high chance that your paper may be accepted in a very high quality journal. So this point I wanted to highlight. That's why it is marked red. Okay. And of course, as the other content of my presentation, what is the trend, or what is the opportunity we people who are there in Andhra Pradesh, the researchers who are working in these areas related to molecular neuroscience, what sort of an opportunities we can get from the presentation from this particular expert? The opportunity what we have is that we can always look for a collaborative research with these sort of expert. So we can always think of working with Dr. Ramanathan sir and do some collaborative projects, or institution like Seven Hills College of Pharmacy, which has all the accreditation and has the autonomous status. Whenever you want to write a funded project, you can always collaborate with an expert like him. Okay, and you can always make it. So it's just an what is suggestion only. And of course, the other opportunity for the other researcher is for a PG project, or for a PhD work, or for your independent research. Whenever you want to do some preclinical work, you can always reach out to these people uh, uh, for the purpose of consultancy research. You can pay them, or you can provide all the necessary chemicals and other things, and get their permission, and then you can also visit that sort of an institutions, and then you can always do a consultancy research for your PG, PhD, or for your independent research activities. So collaborative and consultancy research opportunity is always there to explore from this particular institution or from this particular expert. Of course, this institution is a premier institution in Tamil Nadu, and it also provides an opportunity for the researchers to do their PhD work in their institution. So whoever wants to try to do their PhD, they can always go and approach them, and then they can do their PhD in this sort of institutions. And also, these institution, at least in a year, they regularly conduct some quality workshops related to preclinical research, because they have enough number of expert researchers in this particular area, and they have all the facilities there. And because they are associated with medical college, they also have a good other clinical support available. So you can always try to participate in the trainings, especially hands-on training or workshop programs conducted by this particular institution. So you'll get some idea for your research work. And of course, other than all this, simply at least you can speak to him, or you can speak to the expert from this sort of institution and get their advice, or you can always clarify your doubts in this particular areas wherever they are working. So this is something which I wanted to highlight from sir's presentation. Okay. Now, my second study, or my second observation, is from the second speaker of day one, Dr. Astos Kumar. And when I searched for his profile, I found that he earlier worked with NIPER Hyderabad, and I'm not sure, but I'm sure from Hyderabad he moved very recently to NIPER Kolkata, I think so. But whatever maybe, like, we look into his research point of view, and then I found that from his profile, he is very much interested with the diabetes, and diabetic complications. Especially from his lecture, I understood that he has a lot of publications on diabetic neuropathy, and that's what he was also focusing on that particular day. And he was touching many molecular level studies which are done by their institution, of course, because it's a institution of national importance. So definitely they have all the facilities there, and it's not the facilities available, whether you're utilizing that facility or not, that's the different scenario. So since he has the experience both with NIPER Hyderabad and Kolkata now, so molecular level studies and his expertise was very much useful. And he also has the expertise on peripheral neuropathy, and through his profile I came to know that he has already carried out some projects worth more than 2.25 crores uh, related to these areas: diabetics, diabetic complications, peripheral neuropathy, etc. As an investigator, as a co-investigator in all these capacities. And he was emphasizing one important point on diabetic neuropathy. And he was also speaking about STZ induced condition. And one important point he highlighted is that we need to do some behavioral assessment, and we have to do some functional assessment, and we also need to do some biochemical analysis, and we can also do some protein expression studies related to this particular model. So the point which I highlighted here is, what we do is a researcher who is working for a regular private pharmacy college who doesn't have that much of instrumentation facility, what we do is, when we look for an article from a standard journal, we find mostly they will be of molecular level, something like that, and they might be using some sophisticated equipment, they might be using some genetically modified animals. So there are certain limitations for certain researchers who are working for a regular private pharmacy college, and in some cases in other institutions as well. So whenever we see such articles, we think that okay, they have evaluated these parameters, but I find that we cannot get these animals, we cannot use this many number of animals, or we cannot evaluate these parameters. So what we do, out of the different parameters which we are supposed to evaluate, we restrict ourselves to those parameters which are possible or only affordable. So we limit ourselves to that, and we even don't try to explore whether we can do this other parameters which are very much necessary to prove that activity. Can we try it as a collaborative research? Can we try this as a consultancy work? We never even attempt for that. So here, why these premier institutions like NIPER, BITS, BHU, or all these premier institutions, why they are able to get quality paper? Of course, they have the facility, and they have their expertise. Qualified people are there. Of course, everything is okay. But we also have to find out some possibility. We need to find out some opportunity through some means, by collaboration or by consultancy or by some means. And then we try to do some sort of an analysis which is very much meaningful to that particular activity which we are climbing. So only when we do relevant behavioral parameters, when we do relevant functional assessment, when we do all the required and relevant biochemical parameters, and when we do some protein expression studies, of course with all the limitations whatever we have, then definitely it's not that we have to be there with the NIPER or with BITS or something like that to have a quality publication. From anywhere we can do, whether we are an accredited institution or whether we are a non-accredited institution. If we have the mind, if we have that interest that I need to do some quality work, somehow the way will be there, and we can always achieve that. So the point why I marked here is red is that in order to have a quality research output, we have to do relevant and recurred analysis for each and every type of study which we are performing. And then he also said, though he has done many research work from Naipur, Hyderabad, or from Naipur, Kolkata, still he has also done many activities with the hard plate method and tail immersion test. This doesn't cost much. Of course, there are certain international brands where even this hard plate method or tail immersion test itself is costing a few lakhs. But still, with whatever hard plate method we have in our Indian models, still with that also we can efficiently do. And there are many people who also publish the quality papers. So it's not that everything you do at the molecular level, you can also do with simple in vivo screening methods like hard plate method or a tail immersion test. So this is one another point which I want to highlight from sir's presentation. Okay, and what sort of an opportunities we can have, how the researchers from Andhra Pradesh can benefit from this sort of an expert. Of course, he also, because he is representing the government institution, we can always think of a collaborative or a consultancy research work. And institution like Seven Hills College of Pharmacy, when we engage them for an expert lecture or a webinar for this STTP, we can continuously be with them, be in touch with them, and then try to involve in our research proposals, into our research committees. And it's not just with Seven Hills College of Pharmacy, because they are organizers, I'm telling that. And everyone who is also listening to these sort of experts presentation, we also have an opportunity to go and discuss with these people and then do some collaborative or consultancy research. And of course, many PG students would like to do it from Naipur. And of course, when you want to do some PG or a PhD level work, Naipur as a common institution, as a government institution, it's nowhere a competition for us. It's a part of our higher educational institutional setup. So then always we can always motivate students for PG or PhD work in Naipur. And of course, we can always get expert advice for our research work. And like every speaker at their end or at the beginning or at some point of time, they share their contact details. If at all, if you find that they are very much useful or they can help you in some way, you can always note down and you can always reach out to those people. And there is no harm in asking this sort of expert advice. Okay, so this is my second study from the second speaker of the first day. And now the third study is from Dr. Mahendran Shekhar, who was the third speaker, and he represented University Kuala Lumpur. And I have a very good personal friendship with him. So the research areas which he provides is that he has experience and he has the required skills for extraction, isolation, and characterization of active constituents, mainly from medicinal plants. And he is very much interested in developing formulation of natural products. And he's also, though he's a chemistry person, but I know that he's very much also skilled in pharmacological activities, especially for the compounds isolated from medicinal plants. This is one particular area where most of the pharmacological researchers will be looking for, where I can get the support for isolation of compounds, characterization of compounds. Where is the facility available? Who are those experts who can help me? So he is one such person who can always help you in that particular aspects. And from his earlier presentations, which I have listened, and based on my interaction with him, I know that he has done a lot of achievements related to herbal formulations, and then he has developed many cosmetic preparations, and he has won many awards internationally. Especially, he has got many awards in the Malaysia where he is currently working, and he has also got patents related to these sort of formulations. So, whenever we have an opportunity to listen to him, always whenever he shares that I have a presentation for so and so college on so and so topic, whatever work I have, I keep that work aside and then I try to listen to his presentation, because from every single presentation of him, I try to learn something, because he provides three important features for pharmacology. He gives some insights from medicinal chemistry, especially from formulation of natural products, and he doesn't he doesn't stop with extraction, isolation, or characterization. One special feature about his research work is, after characterization and after publishing his data, most important thing is he converts that into a formulation, or he converts that into a product, and then he tries to get the approval for that as a product. So if he has developed a herbal formulation, he doesn't only publish data, he tried to uh, make it as a formulation, and then he tries to commercialize that. And he has shown many examples of those cosmetic preparations which he has developed, and then he is in the process of commercializing that with the help of their students. And every time he used to say that whatever research output I am bringing, it's all because of my students. So they are the actual people who are involved. I'm just helping them to complete that particular work. Okay, so that is one important thing which we need to learn from him. And when I gone through his profile and what sort of projects he has done, and what is the worth, it's not just about the figure, how much crores he has achieved or something. May not be, the number may not be accurate. It was around 3 lakhs or 27,000 Malaysian ringgits, which he has quoted in his profile. I converted that 3,27,000 odd Malaysian ringgits into INR, and I got this value of around this 6.5 crores or something. Maybe I might be wrong, but he is worth of it. And for the researchers in Andhra Pradesh, what benefit we can get, or what sort of an opportunities we have with him. Now we have an opportunity to collaborate internationally. So this is the best opportunity which he is providing. He is open for collaborative research, of course, because it's also a medical university there in Malaysia. They have their own administrative formalities, but consistently when you engage such people, make use of their expertise, and when your institution also show that commitment that we are also looking forward for a quality research, a fruitful research, then sooner or later we have the chance of collaborating with these sort of international experts. Not only experts, you can always collaborate with the university which he is representing. And for a autonomous NBA NAC accredited institution like Seven Hills College of Pharmacy, it will be much easier for you to approach such a university in Malaysia to collaborate. And he is such a person who is very helpful and who is very much reachable at any point of time to help us. And of course, you maybe also have a good relationship with him, but it's my personal suggestion which I am sharing with you. And of course, for students or for the faculty members, for an independent research or for your PhD research, whenever you want to do, or there are many funded projects where we can collaborate as an higher education institution in India. We can always collaborate with an foreign university. And when you have a foreign university very close to India, and they also have proved experience with that. There is very high chance that our proposals may be accepted. So for PG studies for students, for PhD work for faculty as well as young faculty members, or for their part of their PhD work for senior faculty, or for any independent research, we can always approach these sort of institutions or these experts. And of course, like the other national experts, we can always have them as an international expert, and for many other activities, and we can make use of it. So one important point which I want to emphasize from Sir's presentation is, as a researcher who is doing herbal based research activities, don't just stop with extraction, isolation, characterization, publishing quality papers. Convert it into a formulation, convert it into a product, and try to bring some commercial value. Commercial value means whether it is going to be a very big hit hit formulation or not. It may or may not happen, but you make a sincere attempt to bring out a formulation out of it, and then try our level best to commercialize it, so that when we do it continuously, at some point of time we will succeed in this. And he is such a person who is an example for uh, formulation, development, and commercialization of such formulation. Okay, so this is one important point which I want to highlight from Sir's presentation. And this is the major part of my presentation. How can we find an article, or how can we find potential collaborators? So, of course, we come to know about some people through these webinars. We come to know through our personal contacts. So we get some people. So how much percentage we can say? We can know few people related to our area to a maximum of 20 to 30%. The remaining 70% of the experts, how do we find them? How do we find them? Or how can we find a good quality article related to our area of research? So, as a faculty members, most of you will be knowing about this concept. There is a concept called Boolean search, and something like Boolean operators. And whenever we search for an article, or whenever we search with a keyword, we use the word or we use word and we also use not. So there is something related to it, and I'll try to show it with the help of a demonstration. Okay, instead of saying it clearly, I will just show it as a demonstration in the next 10-20 minutes. So just search for what is Boolean search if you're not knowing about it, how to effectively use this Boolean operators in the search engines, and how to get our data very easily. So just I'm giving the lead, search for Boolean operators. Now, the other important aspect is that we can get the relevant article, or we can identify the potential collaborator. We can identify the potential collaborating institution through certain sources. The first source of getting the articles, identifying the potential collaborators or institutions who are doing this sort of research, both nationally or internationally. The first source is journal search engines or journal search databases. And some of the reputed or most trusted journal search engines or databases are listed, and out of many, we have PubMed, we have Science Direct, we have Scopus, we have Web of Science, we have Springer, we have Ovid, which is from Wolters Kluwer company, and there are many other search engines. We also have something like ResearchGate, we have something like Google Scholar, but these are something which are more reputed, and there can be much better than this also, but at least we can get whatever we want from these sort of journal search engines. Why I have highlighted PubMed and Science Director? Because without institutional logins, without subscriptions, we can directly find articles into this. Some of the articles may be open access, some may be paid one, but at least we can at least get some idea, abstract, or in some cases we can even get a full text article freely downloadable. And the other reason why I have highlighted the first two is that I wanted to practically show to some people how to use the Boolean operators in these two search engines. That's my purpose. And if you are already having an idea how to use this Boolean operators in these set of search engines, it's okay. But I just wanted to give some trial to those people who are not aware of how to use this Boolean operators. And from where else we can get the information regarding the article or a potential collaborator. So we can always look for the company websites. We can always look for the company websites, and we can also visit the institutional websites. Institutional website means you can open Naipur Mohali, see what is there inside. You can look into Banaras Hindu University, you can see what is there in Punjab University. So like whatever premier institutions we know, or whatever pharmacy colleges we know, whether it is accredited, non-accredited. So irrespective of any such things, we can always open and then see what sort of research activity they are doing, which areas they are considering, who are those experts. So we can always look into that. And I have mentioned one word here, CPCSE registered establishment. And some people might have seen this document. There is a PDF document which can be downloaded directly from Google in the name of CPCSE registration list. So what is there? Every single institution or every single organization where an animal house is there, or wherever an animal research is carried out, all the institution or the organization names are mentioned in that particular list. Okay, so what is the usefulness of that particular list? Anyone who wants to do a preclinical research, they can get some information from where actually this preclinical research are happening. You can know about the institutions, you can know about the industries where preclinical research are there. You can simply get that information. And another usefulness of this particular document is it clearly gives you the address also, and it very clearly says where is this company located, because it is an establishment. This establishment will have a very clear address. You cannot mention the corporate address name. You have to actually show where that animal house facility is located. So it's very easy to identify the company's name. You can identify the institution names right from the government one to the private organizations. So we can effectively use this to get an idea. Wonderful source of information for those who are looking for jobs, who are looking for collaborative research, who are looking for experts, very easily obtainable. And of course, other than this list, you can straight away type animal house facilities. You can find out those companies where this preclinical research is being carried out, commercially or non-commercially, and you can always go and see their websites. And I also mentioned the other college because I was also listening to Kumar sir's lecture on the day 2, and I'm just trying to highlight one indication. It's not that I'm showcasing this particular college or I'm highlighting this college, not in that way, because similar to that of the first 3 case studies, this is another one more case study you can take. And the facilities, the expertise available in this particular college also is very much relevant to our theme. And that's why I wanted to highlight. So you can individually see in the college websites or the company website, and then you can get the information, what sort of research is being going on, and what are the products they have developed, what are their research outputs, all those things. And how can we find the potential collaborators. So you can also find a potential collaborator or the institutions where the opportunities are there for preclinical research, even from job search engines. Though you are not looking for job related matters, but still by searching in these sort of job search engines, you can identify where this particular type of preclinical research is happening, where preclinical neuroscience research is happening, where Alzheimer's related studies are happening, what sort of models they are using, what are the qualifications required for them. So these things you can always get from this particular sites. So job search engines are not only for job search, it is also for many other purposes if we make use of these engines very effectively. And most importantly, we have social media, social networking sites, and every one of us are highlighting the importance of LinkedIn. LinkedIn is a very, very effective tool nowadays. Many HR representatives or many HR managers of leading companies, they are looking for LinkedIn profiles. They are looking for LinkedIn profiles. Okay, and through the same LinkedIn, you can always find the potential collaborator. You can see what are all the current trends in your area of research, who are those experts related to that, which are the premier institutions which are doing that. So you can always get all this sort of informations from social media networking sites. So it's not just LinkedIn alone. You can effectively use Facebook, you can use Twitter, you can use Insta accounts, and you can use many other social media tools. It's just a representation which I have given to make sure that within one slide I can show everything together. Okay. And fourth one is something which I said earlier. Fourth content which I want to say is I want to give you a checklist of a research proposal which can be used for preparing a research proposal. And before I acknowledge, I would also like to quickly share for next few minutes. I would like to share some of the important documents. I hope you are able to see the screen. The screen is loading. Is add instruments visible?
Yes, sir.
So you can see this is one of the leading preclinical instrument supplier. You can find one important tool here, something like NIBP system. NIBP system is used for non-invasive blood pressure measurement. This particular device will cost around 3 to 4 lakh rupees. We can use it very effectively for antihypertensive drug screening. And a paper generated out of a study done with the help of NIBP systems from AD instrument or something like this will make your paper more standard, and there is a very high chance that it will be accepted by the reputed journals. Okay, so AD instruments is one of the important thing. And what else you can get from these sort of research instrument companies, okay, or the suppliers? You will get a lot of data regarding publications related to those sort of equipments. So there will be many events which are happening or being organized by these sort of companies worldwide. So when they conduct such quality events in India, they will always introduce this new equipments in those events. So if such events are happening in somewhere in Andhra Pradesh or in Telangana or in Tamil Nadu or in Bangalore, very near to us, try to make use of the opportunity, go and see what is that instrument, how to use that particular instrument, and we can see and observe all those things. And whenever we have a chance, when we know about that instrument, then we can always write a research proposal better when we know about these instruments. Okay, so now the next one which I wanted to share is similar to AD instrument. There is another one more called PanLab or Harvard apparatus, as you can see in the screen. As you can see in the screen. So related to my title, you can see under behavioral research, there are many sub-themes are available, just like video tracking, pain and analgesia, attention and impulsivity, operant conditioning, anxiety and depression. So different themes are there. Now you can choose which one will be more easier for you, which one will be very much rewarding for you. You can select such theme and then you focus on that particular theme and then try to do all your activities relevant to that particular theme. Say for example, when there are these many number of themes available, I am very much interested towards learning and memory. So in behavioral research, though I have an idea about all the other models as well, but I mainly target learning and memory models. So if you click on this learning and memory, there will be many equipments related to learning and memory. Just a quick glimpse I will show you. So you can see what is the current animal models or the names of the equipments which is used for learning and memory assessment. See here, a delayed alternation task using T maze, Y maze, fear conditioning test, Morris water maze test. So what is that information you get from this particular site? Very clearly it tells for evaluating learning and memory, which model is used worldwide. Very clear information will be there. Just if you click on this particular read more, it will give a brief description of what are the parameters to be measured, what is the name of the instrument, what is the design, everything it will give you. And it will also give some reference articles for each and every related equipment or that particular model. And the most important point is these are currently in trend. These are even now used worldwide. Only then this company will market that products. So this is another one more useful information where you can always get it from a preclinical research instrument supplier or a manufacturer. Okay. And when you have free time, when you look into these sort of sites, you get a better understanding about that particular instrument. When you write a research proposal, it will definitely give more meaning to your research proposal. Okay. Now, the next important site, as I told you, PhD College of Pharmacy, and straight away I'm opening Department of Pharmacology. You can see here what are the instruments available with that particular institution. You can see ELISA reader with spectrometer, all this, electrophoresis. So as sir has mentioned in his presentation, they have all the facilities both for in vitro screening as well as in vivo screening. I was explaining about the stereotactic apparatus. I can show it over here, a stereotactic device from a branded company or WD Life Sciences, surgical apparatus, infusion pumps. There are many, many things. And you can see the publications and you can see Dr. Ramanathan sir's, what you'll say, fundings, whatever he has obtained, and what are the funding agencies. What is the usefulness of this particular data? You know, these experts, under which areas they were able to get this particular grant, it will give you an idea. So we cannot simply copy this particular idea. Simply if we copy also, we are not going to get the grant. But at least a researcher, a middle level researcher or a senior researcher, when you look into these sort of titles, you will get to know what sort of projects are generally being approved by different funding agencies. Of course, we should have the expertise to claim that I want this grant. But to get an idea, only when you continuously search for such information, you'll update yourself, and sometime later you will get the benefit. Just a sample. And now regarding Ashutosh Kumar sir, so he is now representing NIPER Kolkata, but his profile was found in NIPER Hyderabad. I'm just showing it. Sometimes it may be wrong, but you can see here, we can come to know about what sort of projects he has got. And see here, as he was speaking, everything is related to diabetes, diabetic neuropathy, neuropathic pain, something like this. So what's that? He is very clear that his area is diabetes and peripheral neuropathy. So he focuses only on two areas and he excels in it. So this is another one more lesson which we have to learn as a researcher. It's not looking into so many areas. Just focus on one or two areas, try to get hands-on skills, and sooner or later you'll become an expert researcher in that area. And now for the University of Kuala Lumpur, Dr. Mahendran Shekhar. So you can see here, so this is from his side. One important information which I wanted to share over here is, see the researcher identity profile. He has created his researcher identity profile in ARCHID, Academia, Myrid, ResearchGate, Researcher ID, Scopus. So these are some of the leading researcher identity. There are many things like that, Publons or other things. For India, there is even a Vidwan site is there. So there are national and international reputed researcher identity profile databases. If you want to create this profile, you should have done some research. Only when you look into the other people, how they have done, what set of research, we'll get motivated. Sooner or later, we also will have a very good profile from this. So this is one important lesson which I wanted to highlight from Sir's profile. Okay. And as I told you, I want to quickly show how to use the Boolean search. So this is the PubMed search engine. Most of you may be knowing, but some may be interested to know. Just go to advanced search and type, for example, stroke, and then select and or not. So for example, I'm showing and. So I have selected the word stroke. So it is appearing here. So it is appearing stroke here. Now I'm searching for the second word. I'm looking for Andhra. Okay, so this is my second word. Now I'm searching for the articles, both for stroke and Andhra. Now you see here in PubMed, there is only four results. And you can see here, all these are full text article because I have used a filter called a free full text. If I remove this option, there will be many more. Likewise, I have filtered the article from 2009 to 2021. Likewise, it's up to you. You can always use whatever tools you can. And I have selected only animal data. So only four are appearing. So now what is the usefulness of this? Now just click on this particular article. You will find a cerebroprotective activity of so-and-so using global cerebral ischemia. Now, what is the other important information? You can find a collaborator who is not in other states. You find a collaborator within your own state. Okay, just expand here. Click expand here. You find a collaborator here. Okay, Department of Pharmacology, University College of Pharmaceutical Sciences, Acharya Nagarjuna University. Okay, so
Excuse
this is
me,
very
sir.
simple. Yes, ma'am.
Yeah.
I'm
Sorry.
getting late.
Yeah, sorry to interrupt you. We have
No problem, no problem.
5 more minutes time for the
Oh, yeah.
So please continue.
So this is one way we can find the potential collaborator. You can find a relevant article and the institution where your area of research is being carried out in your own state or in our own state. This is one of the effective tools. Same thing with Science Direct. There also you can use the same thing by advanced research. And in Panacea Biotech, you visit a pharmaceutical company and then see what sort of job roles are there. I will show you an example here. Like there is one job profile here, pharmaceutical research development. The position called senior executive for pharmacology research. The place is Punjab. So what is that? You know that there is a job opportunity related to neuroscience research in Panacea Biotech. The unit is located in Punjab. So very clearly it shows where all the opportunities lies. What sort of animal models, if you get expertise, you will have a job prospect. This is one simple example for it. Okay, so this is something which I wanted to highlight with people. And quickly, last 1 minute, I will show you that document. So you find a CPCSE registration list. You get to know about all the institutions or organizations where all the preclinical research is happening. You will have a complete address also, so you know where all the opportunities lie. So you can use this particular opportunity. And now, the last part, I want to acknowledge the organizers of this STTP program from Seven Hills College of Pharmacy. And I would also express my sincere thanks to my management, Bapetla College of Pharmacy and Bapetla Educational Society for giving me this opportunity to speak on this particular topic. I hope I have given some of the insights, not as an expert researcher, at least as a researcher who is at the beginning stage. Thank you. Yes, ma'am, I'm done.
Yes, sir. Very wonderful presentation. And I just request you to stop sharing the presentation, sir. Please.
Ma'am?
I request you to stop sharing the presentation.
Okay. So it's done.
Yeah. So I thank you immensely for your invitation, for your accepting the invite, and also for giving a wonderful presentation on how to really look for the seminars and other programs like STTP, and how a researcher wants to be clearly represented, their ideas, and also the collaborations. So thank you so much for your wonderful insights, sir. So I just take this opportunity to present a gratitude of appreciation to you from the management and principal side. So kindly accept the certificate virtually, sir.
Thank you very much, ma'am. Thank you very
and
much.
Also we are sending you a memento for your wonderful talk on this day. So we'll be just sending the post in a couple of a week or two. And thank you so much, sir. Stay in connect with Seven Hills College of Pharmacy.
My sincere thanks to the management of your college, principal sir, all the faculty members who has given me the opportunity. And I also thank all the fellow participants who were listening to my presentation. Thank you.
Thank you, sir. Thank you for diversifying it. Thank you so very much. Thank you, sir. Faculty members, I requested you to stay back in the meeting, and as we are to the scheduled time of 11:30, and we have our today's session speaker of day 2, Professor Divya Ora, ma'am. Ma'am, very good morning to you. Ma'am, very good morning. Am I audible to you, ma'am? Madam, very good morning.
Yeah, good morning, Dr. Sucharita.
Yeah, ma'am.
Yeah, I'm good. I was getting a message that I need host permission to unmute, so I was waiting. Yeah.
Okay, ma'am. Okay, ma'am. Can you please turn on your mic, ma'am, if it is permissible? I think you have turned on right now.
Yeah, I have turned it on now.
Yeah.
So am I audible?
Yeah, perfect and audible, right? How do you do, ma'am?
Yeah, I'm good. Very well. Thank you.
Yeah, thank you so much, ma'am. Thank you. How is the things there? Everything fine, ma'am?
So you want me to start now?
Yeah, I'm just asking you, like, how was the things there? Is
How was
everything
the?
fine? How was
Yeah,
the
yeah.
thing? Yeah, how was the things there? It's going
Things
well,
are
right?
going well. And congratulations to you for organizing such a wonderful FTP program.
Thank you, ma'am. Thank you so
Yeah.
much. Thank you so much. And thank you for accepting an invite to just, I made a call to you over a phone, and with a small word, I just accepted my invitation and been on this day of day 3 of ESTTP organized with St. Hills College of Pharmacy, ma'am. So wonderful and so inspiring, especially for me to see your bio and also to meet other slides, all those things and all. It was so inspiring, actually.
No, it is my pleasure.
Thank you, ma'am. Thank you. So I just requesting all the participants to make use of this opportunity and get the best out of the madam too. And so to introduce this today's speaker, Professor Divya Ora, ma'am. So ma'am is a professor in Department of Pharmacology, School of Pharmaceutical Education and Research, Jamia Hamdard. And to introduce the achievements of Divya ma'am, I request my dear colleague Lavanya madam to do the need. And madam, it's over to you.
A very good morning to all the delegates and the dignitaries. Myself, Lavanya, Associate Professor from the Department of Pharmaceutical Chemistry, and it's my privilege to brief about today's session's resource person, Dr. Divya Vahora ma'am. Ma'am is currently working as a professor in the Department of Pharmacology in School of Pharmaceutical Education and Research at Jamia Hamda, New Delhi. She has over 20 years of teaching and research experience with over 200 publications in reputed national and international journals, and also authorized and editor for 4 books published by Elsevier, CRC Press, and Springer. Ma'am has received grants and worked as a principal investigator for various research projects on neuroscience funded by DST, AICTE, ICMR, UGC, CSIR, and Government of India. She is a coordinator for UGC Special Assistant Program, DST FIS program, and AICTE MODROPS program for her department. To her excellence, 70 research degrees have been awarded under her supervision. And ma'am, to her crown, she is an elected fellow of Royal Society of Biology, London, and she is a recipient of Distinguished Women Scientist Award of Indian Society of Chemists and Biologists, and awardee of Dr. Vinod Kumar Bhargava Award of National Academy of Medical Sciences, and ma'am received Dr. D.N. Prasad Memorial Oration Award of Indian Council of Medical Research, and also
I think that's okay. I can start now.
Ma'am, just a minute, ma'am, just a minute. Please, ma'am, just a minute.
And ma'am has been awarded for Young Teachers by AICTE, Fast Track Award for Young Scientists by DST, and Chandrakanta Dania Prize for Best Published Paper in Pharmacology. Ma'am has been excel, and she is a member of International Advisory Group of British Pharmacological Society, UK, and is an expert member for research grants submitted to National Science Centre, Poland, and La Laxia Foundation, Spain. Her major areas of interest are neurobehavioral pharmacology, particularly epilepsy and epilepsy-associated complications. Apart from that, neuropsychiatric and neurodegenerative diseases, including Alzheimer's diseases, and associated comorbidities, including osteoporosis. And ma'am has identified novel targets for epilepsy and epileptogenesis and addressed several epilepsy-associated complications in her laboratory. And her work identified various therapeutic options for possible exploration as an adjunct to conventional anti-seizure drugs in preventing neurodegenerative process of temporal lobe epilepsy. And so many new models have been developed for secondary osteoporosis in mice, and novel biomarkers for epilepsy were investigated with a focus on aromatase and histamine H3 receptor antagonism. Potential targets. And I'm privileged, and it's a great opportunity for me to introduce ma'am, and a very hearty welcome to you, ma'am. You can please carry on the session, ma'am, please.
Thank you. Thank you, Dr. Lavanya, for a very detailed introduction. And I'm really grateful to the principal of Seven Hills of College of Pharmacy and Dr. Sucharita for inviting me here for this talk. And so I'll just share my screen. So I hope my screen is visible to all.
Yes, madam, it is visible.
So, so now this is what the day 3 of the FTP program. So I'm sure a lot of new techniques and new molecular strategies have already been discussed by other speakers. So I'm not going to discuss my research over here, and I'm not going to discuss the new techniques and their methodology. What I'm going to be discussing is an overview of how to go ahead with the drug discovery for anti-seizure or anti-epileptogenic therapies, and what is the current need in terms of discovering molecules which are acting on newer molecular targets. And because if you look at the anti-seizure drug discovery, now there has been a lot of problems associated with epilepsy, because even though we have a lot of second generation and third generation anti-seizure drugs, still about 30 to 40% of patients are refractory to therapy, and this medically intractable group is not only having continuous spontaneous recurrent seizures, but they are also at high risk for comorbidities which are associated with epilepsy, and they are also at high risk of sudden unexpected death. And therefore, there is a need to identify newer screening methods for discovering drugs which might be beneficial for such patients. So now, if you look at the anti-seizure drug discovery through much of the past century, so you can see that if you look at the period from 1850 to 1980, you can see that only a handful of drugs were available for clinical use, and but if you look at the period of 1990 to 2020, there are so many new drugs which have been discovered. So the earliest drug which was discovered was potassium bromide, and this was discovered mainly through serendipity. So it was a chance observation that potassium bromide protected a certain type of seizures, what we call today as catamenial epilepsy. So potassium bromide was very effective at that time, and that time the anti-seizure treatment was not focusing on tolerability or safety profile, but they were only focusing on how the drug is able to prevent or reduce seizures. So potassium bromide was used for several years. And after that came the phenobarbital, which was again a discovery which was serendipitous discovery. And actually, phenobarbital was already available as a sedative hypnotic at that time. And however, when phenobarbital was administered to the patient suffering from epilepsy as a tranquilizer, it reduced their seizures. So again, it was a serendipitous discovery. And then physicians at that time observed that phenobarbital was even more effective than bromide, and even effective in patients who were not responding to potassium bromide. And even they combined potassium bromide with phenobarbital at that time. So which we can say was the beginning of the rational polytherapy at that point of time. After that came the discovery of, by Merritt and Putnam, discovery of phenytoin. And so this was the first time that an animal model was systematically used for screening for anti-seizure effects. And Merritt and Putnam actually they screened the drug phenytoin for chemically induced seizures in the cat and electrically induced seizures in the cat, and mainly maximal electroshock seizures. And then they observed that phenytoin was able to completely abolish those type of seizures. So first time an animal model was used for the screening of a drug came based on animal model for the first time. So then after that, derivatives of the already discovered drugs by altering the chemical moiety of the existing drugs, like there is mephenytoin, derivative of phenytoin. Then there is primidone, which is derivative of phenobarbital, and so on. So these drugs were discovered. Ethosuximide, which was very effective against pentylene tetrazole-induced seizures, and it correlates with the generalized absence type of seizures in humans. Then the next landmark discovery was of carbamazepine. Carbamazepine, which even today remains a gold standard for treatment of seizures. And so carbamazepine and valproate. Valproate again is a very broad spectrum anti-seizure drug. So while it has multiple mechanisms and it has efficacy against a lot of multiple type of seizures, whether it is focal seizures or whether it is generalized tonic-clonic seizures or whether it is generalized absence seizures. So, and it was also likewise effective in a lot of animal models to show the clinical correlation with these drugs. Then the last, from 1990 to 2020, a lot of new second and third generation drugs were made available. And so you can see that there was a remarkable success in identifying a lot of new anti-epileptic drugs which were having either a better pharmacokinetic profile, or they were having reduced adverse effects, or they were acting on novel molecular targets in the hope that they would be effective against refractory population. However, the main thing was that despite the introduction of second and third generation anti-seizure drugs, there has been no improvement in the therapeutic efficacy of these drugs. So there are still patients who are refractory to therapy. There are still patients who are not responding even to these drugs, which are acting via novel molecular targets. So in terms of therapeutic efficacy and in terms of disease modification, what we call epileptogenesis, so these molecules were not found to be better than the first generation anti-seizure drugs. So now mainly these molecules were discovered based on the efforts of the NIH program. This is a very old program of National Institute of Health, which is known as Anticonvulsant Screening Program. And recently this program has been renamed as Epilepsy Therapy Screening Program. And a lot of new models have been added under this program also, and which led to identification of a whole new second and third generation drug discovery. Now, I must bring to your notice that International League Against Epilepsy had revised its definition in 2014. So, and a new classification of seizure types came in 2017. So, according to the this new classification, epilepsy is defined by any one of the following conditions. Either you have two unprovoked seizures which are occurring more than 24 hours apart, or you have one unprovoked seizure and there is a probability of further seizures. And this probability of further seizures is occurring over the next 10 years, or there is diagnosis of an epilepsy syndrome. So this is the practical clinical definition of epilepsy. And then the seizures were also classified, reclassified as focal onset and generalized onset. Now, whenever you are classifying seizure, you have been studying about whether they are generalized seizures or whether they are partial seizure. So partial term is now replaced by focal. So they are focal onset seizures or they are generalized to the whole brain. And then focal may be further classified into whether a person is aware or he has impaired awareness. And then you can classify them according to whether what kind of motor symptoms you are having or the non-motor symptoms you are having. So actually, if you look at the new classification, it is more realistic, it is more practical, because it is based on the actual, the symptoms actually observed in a patient. And therefore, it is more realistic explanation of what type of epileptic seizures a person is having. Similarly, in case of generalized onset, you have either the motor symptoms, which are tonic-clonic or only clonic or myoclonic or atonic or epileptic spasms, or you have absence type of seizures, which are characterized by eyelid myoclonia and typical and atypical symptoms. And then, as per the previous classification, partial with secondary generalization, I'm sure you must be remembering, you have studied that. So that has been replaced by focal to bilateral tonic-clonic seizures. And then there must be some seizures where you have missed the onset, or it may be unknown whether they are focal or generalized. So they may be classified under the unknown category. Now, just like seizures are characterized, we have a lot of epilepsy types which are characterized. So like we have focal epilepsy and we have generalized epilepsies. And I have not given the classification of epilepsy types because that itself is a very vast area to cover. Because when we talk about epilepsy, it is a group of disorders. And it is not a single syndrome where we have multiple types of epilepsies and epilepsy syndromes. And therefore, the highly heterogeneous nature of the human epilepsy, coupled with the complexities which are associated with human epilepsy, leads to the fact that no single animal model is likely to be perfect when it comes to identifying drugs for epilepsy. And therefore, screening has to be performed in a number of different types of models to actually know the profile of anti-seizure drug, which is likely to be beneficial in a particular type of epilepsy or in multiple seizure types or epilepsy syndromes. So likewise, we have models for seizure states. And we have different models of seizure states. We have models which are depicting a chronic state like epilepsy, and we have models for status epilepticus, which is actually an emergency treatment where you have continuous, more than 30 minutes of continuous seizure activity, and it is also a very life-threatening situation. Then we have models for epileptogenesis, which I'm going to be discussing in my subsequent slides. And then we have models for the refractory epilepsy, which is refractory to currently available drugs. Now we all know about the models, the basic models of screening for any drug for a particular seizure. And the most common type, which we all know, is the maximal electroshock induced seizures. Of course, there are different variation of the same maximal electroshock test in the form of threshold test or increasing current electroshock threshold test. So these electrical models, I'm sure you all are already aware of. And then similarly, we have chemical models, among which pentylene tetrazole has been the most commonly employed screen for drugs which are effective against generalized absence types of seizures. So depending upon the mechanism, you can either use convulsant for antagonizing potassium-mediated inhibitory processes, or you can antagonize glycine-mediated inhibitory processes, or you can enhance glutamate type synaptic excitation in the brain. So we have a number of electrical as well as chemical models which have been used since several years. And then there is a kindling model where you have by electrical methods or by chemical methods, you can use a subconvulsive dose or a subconvulsive current stimulus, and you use it over a period of time for several weeks till there is intensification of the seizure activity, so that the initial subconvulsive current becomes a convulsive current, or the initial subconvulsive dose becomes a convulsive dose. So regardless of the approach which is used, there is a permanent change in the seizure susceptibility in the kindling model. And you can also implant electrodes in the brain of animals, and you can target a specific area of the brain, whether it is amygdala kindling or hippocampal kindling. And this can produce alterations similar to that found in the chronic epilepsy models. So these models are actually used for the initial anti-seizure drug discovery. They are not used in the late stage drug discovery programs. And generally, we know that MES is mainly looking at the tonic extension of hind limbs, which I showed in this slide, that there is a very clear-cut extension of hind limbs, which you know, if any drug is able to protect, and phenytoin shows remarkable effect. It just abolishes the tonic hind limb extension phase in the animal. And this actually led to development and identification of phenytoin, which is one of the widely used anti-seizure drug, and which is efficacious against generalized tonic-clonic seizures. And then the PTZ model is generally basically looking at myoclonic jerks and four-limb clonus, and it has been successful in discovery of trimethadone and ethosuximide. So, and amygdala kindling, again, it induces focal seizures, which leads to disruption of awareness. You can say that it is a model of what, as per the previous classification, focal seizures were partial seizures. So there are awareness and impaired awareness, which has been new terms, but previously they were known as simple partial and complex partial. So this model can induce focal impaired awareness seizures. And now these models are known as clinically validated models. Now, what does it mean to be clinically validated? Why do we call them clinically validated models? Now, for any model, now if you look at the anti-seizure drugs which have been discovered, whether it is first generation, whether it is second generation, or whether it is third generation anti-seizure drugs. So if you see the number of drugs which have been identified based on these three models, so all these models are sensitive to at least one of the anti-seizure drug which is available in the market. Or you can say the drugs which are available in the market, they are sensitive. They have been screened in at least one or two of these models. They have been found to be efficacious. And therefore, they have been known as clinically validated models. Now, for anything to be called as a clinically validated model, you need to consider several factors. And what are these factors? So the first is the phase validity. Now, when we say phase validity, what do we mean? Phase validity means whenever there is similarity in the biology and symptoms between the animal model of interest and the human disease state being modeled. So that means there has been a similarity in the biology and the symptoms between the animal model which has been done. Now, if you look at the MES and PTZ, both these models, they lack phase validity. They do not have phase validity. So now mainly in epilepsy, why they don't have phase validity? Because also of the complexity and heterogeneous nature of epilepsy, and that there are still the pathophysiology is inadequately understood. So which are the primary reasons? However, if you look at the kindling model, it recapitulates many of the features like behavioral comorbidities which are associated with epilepsy and the neuropathological changes. So therefore, you can say that amygdala kindling is better as compared to MES and PTZ in terms of the phase validity. Now, the second very important factor is the target validity, predictive validity. Now, predictive validity means that the clinically effective interventions, they show effect in this model. Now, here it is very important to note that all the three models, they have predictive validity. Because like I mentioned, the drugs which are available in the market, they are effective in either one of these models. So they have very high predictive validity, and therefore they are still continuing as an initial screen for anti-seizure drug discovery. So in terms of predictive validity, these three models are very good. And then the last one is the target validity. That is, the target under investigation should have a similar role as in the disease, as in the clinical situation. Now here again, you would see that MES and PTZ, they lack target validity. Why they lack target validity? Because they have been conducted in healthy animals. So they have been conducted in neurologically intact animals. So they do not lead to the targets being expressed as in the human disease situation. However, we can say that amygdala kindling still possesses some of the neuropathological features which leads to the similar targets being expressed. And therefore, amygdala kindling was correctly able to identify the anti-seizure actions of levetiracetam, which failed in the MES and the PTZ model. So levetiracetam was a drug. It failed in two models, but it came to be highly efficacious in amygdala kindling. And currently, if you see among the newer generation anti-epileptic drugs, levetiracetam is one of the widely used anti-seizure drug, which has efficacy against even seizures which are not responding to the conventional drugs. And levetiracetam is the only drug which is also prescribed as monotherapy. All other newer generation drugs are prescribed as an adjunct to conventional drugs. However, levetiracetam is given both as an adjunct and also as a single drug in the treatment of epilepsy. Now again, however, despite being clinically validated models for a large number of anti-seizure drugs which are currently available, there is a growing concern that the efficacy of second generation and third generation drugs has not improved. And the efficacy has not improved, which could be because of the fact that we have been using the same models over several decades. And therefore, maybe they are able to identify the drugs which have similar, similar profile as the drugs which are already available. And therefore, we are not able to treat those patients who are not responding to the available medication. So therefore, there is a need for more new models which can actually identify and take care of patients who are not responding to the currently available drugs. Now, again, for kindling, it is important to note that seizures are still, evoked, even though you are giving a subconvulsive electrical stimulus and subconvulsive current. But after kindling has developed, animals are still not having spontaneous recurrent seizures. That means you still have to give a small dose, which is able to elicit seizure. However, in epilepsy, what is happening, there are spontaneously occurring recurrent seizures. So that is the drawback of the kindling model. And so it cannot be called as a true model of epilepsy. So what are the characteristics of an ideal model of epilepsy? So when we say an ideal model of epilepsy, it should be able to develop spontaneously occurring recurrent seizures as it occurs in a human situation. And the seizure type should be similar to those occurring in the human epilepsy. So there are multiple seizure types in human epilepsy. So at least the model should be able to reflect that. There should be age-dependent onset of epilepsy. Now, there are certain epilepsy syndromes which occur only in children. So is your animal model able to present that? Then seizures associated with epileptic-like activity in the electroencephalograph, similar to humans. And then the effective plasma concentrations of anti-seizure drugs. Now, it is very important because most of the anti-seizure drugs, they have a very narrow margin of safety. So you have to maintain their plasma concentration level. So whether that level is similar to the in the animal model also. So all these things you need to consider for an ideal model of epilepsy. Now, it has been seen in humans that generally there has been an initial injury, or like if there is a head injury, or if there is a brain trauma, or if there is a CNS infection. So generally it has been seen that there has been an injury to the brain, and after the injury there is a latent period after which there is development of epilepsy. So the latent period is termed as epileptogenesis. So therefore, a true model of epilepsy should be a true model of epileptogenesis should be able to show a latent, an initial precipitating injury, a latent period, and then the development of spontaneous recurrent seizures. And then, as per International League Against Epilepsy, the pharmacoresistant epilepsy is defined as the one which is not responding to at least two of the adequately tolerated anti-epileptic drug regimes. So therefore, the ideal model of pharmacoresistance should display a profile that is refractory to at least two of the currently available anti-seizure drugs. Now, so coming to the epileptogenesis, so like I already mentioned that there has been an initial, there has been evidence of an initial precipitating injury. So epileptogenesis can and then which is followed by a latent period. And this latent period is then preceded by spontaneously occurring recurrent seizures. Now, this initial precipitating injury, how to induce that? So a very common method has been used is that you induce status epilepticus in the rodents, which causes a brain injury, and continuous seizures damages the brain. And after status epilepticus, you can study the latent period, which is the period of epileptogenesis. So here, now there are certain molecular and cellular changes that you can study during epileptogenesis. There is, neurodegeneration, there is neurogenesis, and there is gliosis, a lot of axonal damage and the mossy fiber sprouting. There is blood-brain barrier damage, there is neuroinflammation, and so on. So all these changes you can study during this latent period, which led to development of spontaneously occurring recurrent seizures. So that means if you are inducing status epilepticus in the animal, so you can study the effect of your drug at multiple time points. So if you are administering your drug with the insult, so it would result in the initial insult modification. However, if you have already induced status epilepticus and after that you are giving your drug, so you can study the period of epileptogenesis, which is nothing but transformation of a normal brain to an epileptic brain. So here you have the opportunity to study all these molecular and cellular changes which are taking place, and you can study the effect of your drug on the development of epileptogenesis. Then you also have the opportunity to study your drug after epileptogenesis has occurred and after epilepsy has occurred, and you can study the effect of your drug on the chronic state, which is epilepsy. So you have the window of opportunity to study your molecule at several time points. So timing would determine the outcome. Now, so here again, like I said, status epilepticus has been used to induce a model of chronic epilepsy state, which is a temporal lobe epilepsy. Now, temporal lobe epilepsy is the most common type of drug refractory epilepsy in humans. And so temporal lobe epilepsy can be induced either using the kynic acid model or the pilocarpine model, or you can also use lithium pilocarpine model. Lithium has also been used. So basically, kynic acid is an analog of glutamate. It acts on the kainate receptors and it produces sufficient excitation in the brain. And pilocarpine is a muscarinic receptor agonist, which has also been used to induce status epilepticus in rodent models, both rats and mice. And lithium has been used to increase the susceptibility of pilocarpine model in the rats. However, it was recently shown that lithium does not enhance the susceptibility of pilocarpine in the mice. It only enhances in the rat. So therefore, mice models generally use pilocarpine alone. However, in the rat model, lithium has been combined with pilocarpine. So initially, when you can induce status epilepticus using any one of these models in the rodents. And you can observe various seizure stages using a scoring system, which is known as Racine scale. And generally, if you have a stage 3.5 and above, then you can say that animals are exhibiting status epilepticus. And you can score them and classify them, whether they are mild or whether they are severe. And you can also do video EEG monitoring simultaneously. And then you can study various histopathological changes. You can also look at the mossy fiber sprouting, where there is, enhanced mossy fibers in the brain. Now, mainly, whether it is kynic acid model, whether it is pilocarpine model, they are mainly damaging the hippocampus region of the brain. And it is very easy to study hippocampal regions, CA1, CA3, and dentate hilus region of the hippocampus. So you can study these changes in the animal, and they resemble to what has been experienced in the temporal lobe epilepsy. So after, say, 72 hours, you can see some changes. After 24 hours, you can see. And you can also study after 4 weeks or 6 weeks, when actually the temporal lobe epilepsy has fully developed. In our lab, we also observed a stage of circling behavior. So we are again now modifying the Rasain scale as per the stage, additional stage which has been observed. And there are several models of status epilepticus and temporal lobe epilepsy, and we have reviewed it here. So we have done a few studies on the kynic acid model as well as on the pilocarpine model. So this is just to show that how kynic acid is actually causing pycnosis in the dentate hilus region of the hippocampus. So it is very prominently visible from the histopathological slide, and you can see that similar degeneration has also been seen in the animal, in the human temporal lobe epilepsy. So, so now there are various methods which can induce epileptogenesis in the rodents. Now, we have already discussed kindling model and the post-status epilepticus model. So kindling model and post-status epilepticus models, they have been used to study epileptogenesis. They have been very widely used to study epileptogenesis, but as I mentioned, that still the drugs identified through these models have been refractory to a large number of patients. So the current need is to identify more models which are etiologically relevant. And therefore, now people are using traumatic brain injury model or the brain infection model. In the brain infection model, they are using, because viral infection enhances the risk of seizures. So they are actually using a virus in susceptible mouse strains, and they are looking at the effects on seizures. Then people have also used cortical dysplasia model. Now in the cortical dysplasia model, this is methyl-deoxymethanol. This is an agent which, if it is administered in utero to the animal, then it is able to show cortical malformations in the animal. So you can induce cortical dysplasia model, and actually cortical dysplasia causes the kind of seizures in humans which is medically intractable to therapy. So you have the opportunity to induce the seizures which are actually occurring in human situation, like post-stroke seizures. So you can use a middle cerebral artery occlusion model, and then also you can study effect on seizures. Or you can also induce seizures using hyperthermia in the neonatal rats, because febrile seizures in the neonates are very common. So I would say that these are more etiologically relevant models, which would be more relevant to the human situation, rather than, of course, post-status and kindling models have been very successful, and they have been successful in identifying a number of drugs. But these models, in addition, should also be a part of the screening curriculum. Then there are models of genetic epilepsies which have been used, but they are they are time-consuming process, even though they exhibit spontaneously recurrent seizures. However, they are expensive models, and then there is a long latency to seizures which are observed, and you have to see that. But zebrafish models are now upcoming, and they have been used by various researchers, and there have been certain mutant strains of epilepsy syndromes, like, for example, Dravet syndrome. Now, Dravet syndrome has a mutation in the SCN1A gene. Or there is another type of epilepsy syndromes which have been successfully studied in the zebrafish model, which are now easy to maintain, and they are less expensive, they are cheaper, and they resemble genes. Most of the genes are similar as in humans. However, some work has been done on the PTZ model, though the consistent reports are still not there. Neurogenesis has been studied following traumatic brain injury in the zebrafish model. However, the same has not been studied as part of the epileptogenesis. So we still have limited understanding of these models. However, these models are coming up, and they might be more etiologically relevant as compared to the models which are already available. Then we also have mouse models of tuberous sclerosis complex. Now, tuberous sclerosis complex is actually the kind of epilepsy in which you have growth of cells in the in a particular region, tuberous sclerosis complex, which leads to continuous spontaneously occurring recurrent seizures. So there is a TSC1 mutation which can be induced in the mice, and these models have been used to identify epileptogenesis. However, the main drawback of these models is that there is a long latency period before actually spontaneously recurrent seizures occur. And like, for example, if you are undergoing a very tedious procedure of middle cerebral artery occlusion, you do not know how many of these animals are actually going to exhibit seizures. So the rate of exhibiting seizure is less as in the human situation. Like, for example, say how many what percentage of patients have stroke induced epilepsy. So that way, these models are not amenable for high throughput screening. However, you can use them to make you can use them to target that 30% of the population which is not responding to the currently available anti-seizure drugs. So in an attempt to make one such etiologically relevant model, we made a very simple, we developed a very simple model of neuroinflammation mediated seizures. And there has been recent evidence of a lot of glial involvement and neuroinflammation in epilepsy. And so what we did was we used lipopolysaccharide, which acts on TLR4 receptors, and it induces inflammatory responses. Pilocarpine is already widely used to induce status epilepticus via acting on M1 receptors, and it causes seizures and neurodegeneration. So we combined these two to develop a model of neuroinflammation mediated epilepsy. And this is a chapter which is published where we have also shown in the hippocampal HT22 cells that how addition of lipopolysaccharide to pilocarpine leads to molecular and cellular changes. So a very interesting observation that we had when we combined LPS with pilocarpine. Now we know that sodium valproate and carbamazepine and levetiracetam, they generally protect against pilocarpine-induced seizures. So when we combined LPS with pilo, sodium valproate and carbamazepine, they lost their efficacy. So that means even though they were effective in reducing seizures against the pilocarpine model, once lipopolysaccharide was added, these drugs did not show their therapeutic efficacy. So we studied the effect of levetiracetam, which showed partial efficacy in this model. So we have recently used this model, and we also observed various histopathological changes in the different regions of the brain, CA1, CA3, and dentate hilus region, where the maximum pycnosis was observed in the LPS plus Pilo model when it was actually combined. And clinically too, if you see that systemic inflammatory diseases, they have been listed in the etiology of certain types of epilepsies. And therefore, you can say that peripheral inflammation can also contribute to seizures. So therefore, we used LPS to induce seizures and to mimic a condition where epilepsy is comorbid with inflammation and where it has both inflammatory component and seizure component in a disease. Now, as I mentioned, that seizures are refractory in 30 to 40% of the patients. So we need to have models which can identify drugs which are effective in pharmacorefractory epilepsy. Now, so 6 Hertz psychomotor seizure model has been used for identifying drugs which are effective in therapy-resistant focal type of seizures. So now, if you compare the 6 Hertz model with the maximal electroshock model, so you can see that 6 Hertz model is working with a low current. And maximal electroshock model is working with a high frequency current, which is 50 to 60 Hertz. However, if you look at the duration, now the 6 Hertz model has a very long duration, while that is 3 seconds. But in maximal electroshock, it is only 0.2 seconds. So you can say, so that means if you use a low frequency but a long duration current, then it is able to identify drugs which are effective against refractory type of seizures. So here you can use corneal electrodes. Here you can use both corneal and ear electrodes. And this mainly induces generalized tonic-clonic type of seizures, while here you can induce focal seizures which resemble psychomotor or limbic seizures in humans. So this has been used as a model, and different features can be induced, automatism, stand position. Now, 6 Hertz seizure model has been used using different currents. Yeah. So it has been used using different currents, like people have used 22 milliampere, 32 milliampere, and 44 milliampere. So now when you use 32 milliampere, that is 1.5 times the CC97, so then majority of the anti-epileptic drugs, they lose their efficacy. Now, and when you use 2 times the CC97, then there is protection given by only levetiracetam, valproate, brivaracetam, and patisigonil. And there is resistance to all these drugs here also. I'm sorry about this error. It should be copied here. So that means if you are using at a higher current, most of the anti-seizure drugs, they lose their efficacy. So therefore, it is used to identify drugs which are effective in therapy-resistant focal type of seizures. So again, levetiracetam is there, valproate is there, brivaracetam is there, which have been found to be efficacious in this model, and so they can be effective against seizures which are not responding to the conventional drugs. And we have used the 6 Hertz model in our lab, and where we showed that when you used a depleter of either brain noradrenaline or serotonin depleter, so that enhances the susceptibility to 6 Hertz psychomotor seizures in the mice. And then we studied the effect of valproate on the same. So valproate is able to show protection when you are using along with levetiracetam and rivaroxaban. So now this is just the decision tree for anticonvulsant drug screening program, which I told you that National Institute of Health anticonvulsant screening program has identified a number of second generation and third generation anti-seizure drugs. So if you look at the anticonvulsant screening program and the decision tree, if a drug has been found to be inactive in the MES-PTZ test, then it is tested in the 6-Hertz psychomotor seizure test. So that means this has been used as an initial screen along with your maximal electroshock and your pentylene tetrazole test. And only if a drug is found to be efficacious in one of these tests, then you can perform kindling, or you can perform the status epilepticus models, or you can go for further advanced studies, mechanistic studies, and so on. So 6-Hertz model is now considered a very useful test, which has been included in the initial drug screening programs. Now coming to the models of drug refractory epilepsy. So there has been certain models of drug-resistant epilepsy which people have used. I have already discussed the 6-Hertz psychomotor seizure model in mice, where at 44 milliampere there is resistance to several anti-epileptic drugs except these drugs. So now there have been, people have used other types of models, like one of these model is that where they have pre-treated kindled rats with a low dose of lamotrigine. And when you pre-treat with a low dose of lamotrigine, then the resistance occurred to lamotrigine as well as to other anti-seizure drugs, which are sodium channel blockers. So this model has also been used. So while inducing kindling, you are treating with a low dose of anticonvulsant, which is leading to development of resistance. Though mechanisms associated with this have not been studied so much in detail. And then I have already discussed the MAM exposed rat, that is methyl zoxamethanol, which if you are administering them in utero, so they can lead to a lot of cortical malformations, and this can cause resistance to ethosuximide, valproate, and carbamazepine. So you can use it as a model which is resistant to ethosuximide and valproate, which is otherwise effective against a large number of multiple seizure types. Then people have also developed phenytoin-resistant kindled rat model. So phenytoin-resistant kindled rat model, but again, this is very time-consuming. It requires a very large number of animals, because sometimes when you are studying the efficacy of phenytoin against amygdala kindled rats, then a certain percentage of animals are not responding to the effect of phenytoin. So you have to choose responders and non-responders. So now again, there is a very less percentage of non-responders, and those are considered naturally resistant to phenytoin. So you select the non-responder population, and then you can screen the effect of your anti-seizure drugs on the same. So, and this resistance has been extended to various other anti-seizure drugs also, except levetiracetam. So people have studied multiple models for studying drug resistance, and also post-status epilepticus models of temporal lobe epilepsy in rats, where again responder, non-responder selection. So you have to segregate animals, which is again, you require a large number of animals, which is practically not possible if you are going to screen a lot of drugs. So these models have been shown to have be as drug-resistant models, and 6-Hz psychomotor seizure is the most simple screen among the models which I have shared. So now I have discussed most of the animal models which have been used, and I have not shared the genetic models because there are so many genetic models, and that itself is a presentation in itself. So, however, there are genetically models are also different types of models where you can either use those animals which are already genetically epilepsy prone, or you are using models where if you give a slight provocation, they induce seizures. So there are different types of genetic models also. And then whenever you are screening for any anti-seizure drug, you also have to see the various adverse effects on the motor and cognitive functions. Now, so because most of the anti-epileptic drugs, if you can say that one of their adverse effect is ataxia. And so impaired motor coordination is there. There is cognitive dysfunction. Most of the anti-epileptic drugs, they impair cognitive functions. There is seizure-associated depression. Most of them are hepatotoxic. They either induce or inhibit cytochrome P450 enzymes. So therefore, they have interaction with a large number of drugs, which is clinically a problem. And then they have adverse effects on bone. So in our lab, we have studied a lot of effect of anti-seizure drugs on bone loss. And we have also developed a model of phenytoin-induced bone loss after titrating the dose of phenytoin. And so a lot of drugs have been found to cause bone loss in both normal as well as over-optimized animals. And so these studies we have been carrying out. We have a bone mineral density tester, and we also evaluate bone biomarkers in different types of bones. So now coming to the testing of newer compounds. So this is actually the overall models which have been used. So of course, in vivo invertebrate models are now coming up. Drosophila has not been very consistent because Drosophila, there is a lot of contradictory studies that show efficacy of different anti-epileptic drugs. However, zebrafish is now coming up as a good model where a lot of scope is there to induce mutant strains. Now I also had a chance to listen to the previous presenter for some time, where the importance of in vitro models was highlighted. So here I would like to bring to your notice that epilepsy, I think, is the only condition where in vitro models are not used in the primary screen. So generally your screening starts, whether it is NIH screening protocol or whether it is all over the world. So screening generally starts with these tests, even though in vitro models have been used, but the seizure-induced electrographic changes you are not able to study in that. And generally the in vivo models are considered only reliable model for identifying drugs which would be found to be effective in human type of epilepsies. And of course, you need to also conduct studies on neurobehavioral adverse effects, which is very important because most of the anti-seizure drugs, they have a lot of adverse effects. However, in vitro models, you can now study brain slices from human resected tissues and of drug-resistant epilepsy patients who are going for surgery, and that has been found to be important in studying mechanisms and other things related to drug refractory type of epilepsy. Now, recently we have published a volume of neuromethods under STRINGER protocols, which is experimental and translational methods to screen drugs effective against seizures and epilepsy. And a lot of contributors are there from all over the world who are expert in epilepsy research. So anybody who is interested in any of the chapter related to these models, they can just email me and I will share the PDF with them. So finally, what is the path from discovery to cure in the future? So now, as I said, that resistance is the main issue in case of epilepsy. So you need to identify the etiology, the cause of epilepsy, which can be either structural, which could be metabolic, which could be genetic, which could be immune mediated. So there is so much of heterogeneity in the different types of epilepsies. There are epilepsy syndromes in which you have several types of mutations. So the path from discovery to cure would actually, including when you are screening or when you are developing an animal model, you first need to identify the cause and correlate it with the human situation. And then you need to study the development of epileptogenesis, so that because 40% of the epilepsies are acquired epilepsies. So can we find a drug which can prevent the development of epilepsy? So the drugs which are studying against the whole process of epileptogenesis would be really beneficial in preventing the risk for patients who are at risk after, say, a brain injury or after a trauma or after a stroke or after any such precipitating factor. So studying epileptogenesis is very important. And then you can also study development of spontaneously occurring recurrent seizures. Once you have developed the model of epilepsy, you need to study the effect of drugs on these models, because our 30% of patients are refractory to therapy. So we need to identify drugs which are effective against epilepsy and can cure epilepsy. We don't have any drug which is neuroprotective, which can actually act on, except there is some evidence for valproate. However, in human situation, this has not been found to be very efficacious. So you need to complement your existing models which have been developed with more of the chronic models or more of the etiologically relevant models. Then you can also develop more syndrome-specific models and also the drug refractory models. And then only if you complement with the existing models, of course, many of these models are not amenable to high throughput screening. So you don't have to include these models in the initial screen. The initial screen would be limited to your 6 Hertz or which is able to identify drug refractory cases also. And then you have to proceed with these chronic models and you have to perform mechanistic studies and so on. So in the end, I think I would like to thank the funding agencies which have supported our lab, UGC SAP DRS 1 and 2, DST-PIST program, AICTE MODROPS program, and ICMR. And I've quoted the work of some of my students who have already done PhD in this area. We have certain collaborations with AIMS New Delhi where we get patients. So like, for example, I mentioned about a lot of anti-epileptic patients, they experience bone disease. So what we did was animal studies were done at our institution, and then we collaborated with them and they gave us patient and we studied the biomarkers in the patients who are suffering from epilepsy. And then the National Institute of Nutrition, Hyderabad, we initially collaborated when we did not have a DEXA scan. Now we have a DEXA scan of our own. And CTRI Lucknow also, because that is for expertise in the area related to bone diseases. So I thank you all for the patient listening, and I wish everyone to remain safe and healthy. And I thank you for giving me this opportunity. Thank you. So if there are any questions, I would be happy to answer.
So nice of you, ma'am. Great to hear such a wonderful speech, actually. And if you permit me, If you permit me, I'll take a few more questions in the line. No?
Yeah, sure.
Yeah, ma'am. There is a question from Dr. Vangara Anirudh. So, respected speaker, does all the marketed drugs qualify all tests like face validity, predictor validity, and targeted validity?
No, like I mentioned earlier, that no model is perfect, and the drugs which have been marketed already, so they do not qualify for, because if you see majority of the drugs, they are not protecting against seizure. They are only protecting against seizures. They are not neuroprotective. They are not working on stopping the disease progression. They are not preventing the disease neurodegeneration. So so they are only most of the drugs have only been clinically validated as far as predictive validity for seizures are concerned. However, the disease modification is not addressed by any of the second or third generation drugs, though there has been some evidence for valproate being neuroprotective, but that is also controversial. And we do not have currently any drug which can actually stop the process of epileptogenesis. So target validity and phase validity are very difficult for a disease like epilepsy, which is highly heterogeneous in nature, and no single drug is able to protect against all the type of seizures or all the etiologies or all the different types of epilepsy syndromes. So therefore, that is the issue, that one has to work on identifying molecules which can be neuroprotective in addition to having protective effect against seizures.
Yes, ma'am. Like you said. And I have one more question, ma'am.
Yes.
Duration, efficacy, and process to qualify all tests like face validity, predictor validity, and targeted validity.
No, I didn't get you. Can you repeat?
Duration, efficacy, and process to qualify all tests like face validity, predictor validity, and targeted validity. He's just asking duration, efficacy, and process to qualify.
Duration.
Yeah, duration, efficacy.
Like how much duration is required for a drug to pass. So again, I would say that because already the drugs which are available in the market, they are not fulfilling this criteria. So this is what researchers have been working on. They have been trying to identify drugs which can, and they have been studying various durations. So it depends on the animal model used and the efficacy against seizure types. If you are asking efficacy in general, I will not be able to give you answer to that, because as far as animal models are concerned, every drug has different efficacy against different seizure types, against different etiologies, against multiple syndromes. So you cannot generalize to have one answer to this question. However, nowadays, a lot of, like I mentioned about the etiologically relevant models, which are being researched, and that might answer you how you can develop, like, for example, if you are focusing only on the Dravet syndrome, which has very medically intractable seizures. So you can use the SCN1 mutant animals, and you can study the effect of drugs, and then you can see whether it is fulfilling the criteria of face validity, predictive validity, and the target validity. However, you cannot generalize it for any other type of seizures. Like if you are studying generalized absence seizures, the duration efficacy would be different. So you cannot generalize, because like I said, epilepsy is highly heterogeneous and highly complex in pathophysiology. So this cannot be generalized and say that this duration, if you study, or this efficacy you study, it would fulfill the need for all different types. So every drug has a different profile. However, recently the new addition was cannabidiol. Cannabidiol was the new anti-seizure drug which has recently been identified, and there they have used how it is identified, because they have complement the existing animal model with more of the etiologically relevant models, and that is how cannabidiol was found to be effective in pediatric epilepsies.
Thank you, ma'am. And the participants are requested to post the queries in the chat box, or you can have a permission to unmute yourself and you can make use of this opportunity. You can straight away talk to the Divya ma'am and she can better answer all your queries. So is there any queries from the participants?
I would be very happy to take any further query right now or in email, whatever you feel comfortable with. I forgot to put my email on the slide, so you can ask Dr. Charita ma'am to give you. So you can always contact me for anything you need.
Yes, ma'am. I'll share to all our participants too. And as you said, the interested participants, I'll just let you know, ma'am. You just share the book details, all those things to them also.
Sure.
So
I'll do that.
Yeah, yes, ma'am. Yes, ma'am. And so grateful to you, ma'am, actually. And thank you for accepting the invite and done the best, as you said.
Thank you. It's my pleasure.
Yeah, and just take this opportunity to present a small certificate of appreciation to Divya Ora, ma'am. Ma'am, kindly accept it virtually, ma'am.
Thank you.
And I'll be just posting this to you in a week or two.
Thank you.
Just a moment, ma'am. Just a moment. I think Sunil, madam left, right? Sir, Dr. Tapa, sir, we couldn't able to get an access to madam. I think madam left, it seems. Yeah, sorry, Tapa sir, we couldn't able to get an answer for your question, and definitely I'll just mail these questions to madam, and I'll find this answer for all those questions to you, sir. Sorry, because madam is not in the meeting, and all the participants are requested to join for the afternoon session by 1:50 a.m., 1:50 p.m. sharp, and thank you so much, one and all.